So I would like to introduce our next speaker, Emma Jones, a PhD student from the Institute of Medical Research and Physics Department, University of Cambridge. Emma uses novel imaging approaches to understand molecular mechanisms of life. Her title is Characterizing the Molecular Interactions of Malaria Parasites Invading Red Blood Cells in Dynamic Conditions. Please welcome her.
Hi, so today I'm going to be talking about malaria. This is a devastating disease which in 2021 caused over 600,000 deaths. This was predominantly in sub-Saharan Africa in children under 5 and pregnant women. The life cycle of malaria is incredibly complex, but all of the clinical symptoms are caused by the blood stage of the infection. This causes febrile episodes, body aches, headaches, nausea, and general weakness. And in severe infections, it causes severe anemia and organ dysfunction. The blood cycle is a 48-hour cycle in Plasmodium falciparum, which is the most deadliest strain of malaria. And in this cycle, the late-stage infected red blood cells aggress to release 16 to 32 new parasites called merozoites. These go on to invade new red blood cells, and then they go on to develop over 48 hours into a new schizont. So I focus on the step of invasion, which is a key point for targeting with treatments such as vaccines. And it's an incredibly rapid process. You can see the infected schizont here is about to aggress. It releases the new merozoites, and they go on to attach to the red blood cell, deform it, reorientate, and then they push into the red blood cell. And this process is incredibly rapid. This video here is shown in real time, and the invasion process takes less than 10 seconds. So traditionally, how we've studied this is we've done endpoint assays or we've done video microscopy. And these are, the majority of the time, done under very static conditions, but these are a poor mimic for what actually happens in the body. In the body, this whole invasion process is happening in the blood circulation, where everything's moving and interacting differently to in a static Petri dish. So in my PhD project, I've looked at using new methods to further our understanding of invasion. I've used optical tweezers to understand the molecular interactions of attachment between the parasite and the red blood cell. I've used different shaking conditions as a way of just introducing more dynamic conditions, and this is quite a high throughput assay. And then I've done a much lower throughput assay, which gets us much closer to the blood circulation by developing a microfluidic device to follow the invasion process. So starting with the optical tweezer assay, what we do is we video the schizont as it aggresses. We use the optical trap to pick up a merozoite and put it on a new red blood cell. We then bring in a second red blood cell with the optical trap and allow it to attach, and then we pull the whole thing apart. So this is it live. You can see here's the merozoite, here's the second red blood cell. Attachment is occurring here. And then once it's attached, you turn back on the optical trap and you pull it off. And you will see here that the red blood cell is deforming quite significantly when this happens. And because we understand the mechanical properties of the red blood cells very well, we're able to use them like tiny force meters. By measuring the amount of deformation that has occurred, we can calculate the force with which the detachment happened. And then what we can do is we can look at a range of different molecular ways of perturbing the system, using genetically modified lines and different inhibitors. And we can use this to target the different proteins of the invasion process. And these are all candidates for potential vaccines. So I started off with using a genetic disruption of this protein MSP1. And this was our best candidate for what was involved in the initial attachment. And very interestingly, I didn't see a significant difference in detachment force, ruling out that it's probably actually involved in this step of the invasion process. I then looked at 2 families of proteins involved in the deformation stage, the EBAs and RHs, and found that several of these proteins did cause a significant reduction in the detachment force. So this points towards these being the proteins that are generating the strength of the interaction between the different cells. I then looked at proteins involved later in the invasion process, and I found that, interestingly, these actually tend to increase the detachment force, likely because they're trapping the parasites in the strong deformation stage. So then, moving on, I wanted to find a way of which we could assess the invasion process under more dynamic conditions. And I started off doing this using a high throughput shaking assay. What we knew from the literature already was that when you shook really fast, you increased the invasion rate, but no one had ever looked over a range of different shaking speeds. So when I did this in 3 different wild-type lines, we saw a really interesting and unexpected behavior. We saw that between static and a very low shaking speed where the blood isn't being perturbed, there was very little change. But then when you start increasing the shaking speed, you actually decrease the growth rate of the parasites. This hasn't been shown before, and it shows that we really need to investigate further the invasion process under these conditions. And then we saw the expected increase in the shaking speed. But the limitation of this assay is when you shake your culture, the fluid is moving, but in order to model and really understand what's going on is incredibly difficult, and it's very variable across the Petri dish. So this isn't really a good mimic for what is happening in the body. It tells us there's something interesting going on, but what we really need is a device that is closer to a blood vessel. So what we then did was we developed this microfluidic chip in which there's a very long channel that snakes backwards and forwards, and the dimensions of this and the flow rates are comparable to where the invasion is occurring in the circulation. So then we take very, very synchronized parasites, and we inject them into our device, and they're treated with a drug, which means that when we wash it off, they'll all start aggressing in our channels. And then once the parasites are released from the infected cells, they'll go on to invade new red blood cells under the controlled flow. We then stain the parasites with a fluorescent dye so that we can visualize them in bright field. And we use image analysis to identify the different cell types in our channel and track their motion as they move along the channel. So if this assay works well, what we'd expect to see is a very constant population of red blood cells, a drop in the population of schizonts as they aggress, an increase in the population of merozoites as they're released from the schizonts, and a corresponding increase in the population of rings as these are the newly invaded forms. So the assay is now working really well. We're able to track all the cell types as they move through the channels. And we see the expected changes in the different population, constant population of red blood cells, drop in the population of schizonts, and an increase in the population of merozoites and rings. And from these numbers, we're therefore able to work out the invasion rate of the parasites. So then what we need to go on to do is understand the invasion under different flow rates. So I've showed that the EBA and RH proteins are responsible for the strength of the interaction between parasites and red blood cells, that the relationship under different shaking speeds is complex, and that we now have an assay to investigate invasion under flow. I'd like to acknowledge my 2 fantastic groups, the Rainer Lab and the Chikuta group, as well as Viola, Morton, Boyko, and Deanne, who have helped develop the microfluidics assay.